Hello dear students, and welcome to my third live biochemistry lecture. The topics for today will be nucleotides and nucleic acids. Let's start with some definitions. What are these bases? Nitrogenous bases in our cells are 5 main types. These are 3 pyrimidine bases, which are uracil, thymine, and cytosine, with 6-membered ring, and 2 pyrimidine purine bases with 9-membered ring. Actually, there are 2 rings. The first one is 6-membered and the second one is 5-membered, but the total number 9. These are called main bases which are involved in DNA and RNA. Except these main bases, we have some other bases that can be formed during the metabolic processes. For example, purine bases that accept adenine and guanine, we have 2 more bases which are hypoxanthine and xanthine. Hypoxanthine is an intermediate, both of them are intermediate in the purine degradation. From hypoxanthine goes to xanthine, and after that xanthine is converted to uric acid. Uric acid is the final product of purine degradation. And do you know if there is high level of uric acid, which is this disease? It's gout. The name is gout. To summarize, all these are purine bases, but these ones, hypoxanthine and xanthine, are not involved in DNA and RNA. They are just produced during the metabolic processes. Another example, there are about pyrimidine bases too. Except these well-known bases like cytosine, uracil, and thymine, there is one more, orotate. The orotate is a part of synthesis of pyrimidines. It is produced as intermediate in pyrimidine synthesis, but again, is not involved in DNA or RNA. Other examples of purine bases, students, we can find in drinks and food. Why is important to know? Because of the disease gout. When we're talking about the gout, we said that this means high level of uric acid in the blood. But to decrease the quantity of uric acid in the blood, for the first level of treatment is diet. Diet means to reduce the purine bases in the diet. That's why you have to know where are purine bases in the diet. In our diet, purine bases can be involved, can be found in some drinks, like coffee. This is the caffeine molecule. In the tea, this is the theophylline, which is absolutely purine base, and in the structure of theobromine, which is involved in cocoa and chocolate, different products from this. Remember that all these are purine bases, which means if there is gout, we need to reduce all these drinks in our diet. One more, where we can find in the food, where, in which food there are purine bases. Because we need to know that to reduce some food if we have gout. Students, actually, if you eat any animal products, meat, in all of them there are nucleus, right? In the nucleus there is DNA or RNA. That's why with meat we can take additional quantity of purine bases, which means if there is gout, reducing the meat in the diet. Modified bases in DNA, the other name is minor bases. What is this? DNA contains small amount of chemically modified bases, the other name is minor bases, such as methylated cytosine. Look here, this is the base called cytosine, and after the methylation process, it can be methylated to form 5-methylcytosine. What is the functions of modified bases? Number one is about the recognition of oligonucleotides, because modified bases help enzymes and proteins distinguish between DNA segments, for example, the self bases from foreign bases. Number two, about the regulation of RNA half-life, because methylation can influence how long RNA molecules persist in the cell before degradation. And the third reason is about the protection, because methylation of DNA protects it from degradation by enzymes restrictases. The methylation most commonly is about two bases. It's cytosine that we said, and guanine is the second base that can be methylated most often. Remember this: high methylation, low gene expression, and one more about epigenetic regulation. Before that, something about epigenetic. This science studies that many factors from environment can influence the genome. One of these factors is a diet, by the way, can influence the genome too. And DNA methylation and histone deacetylation leads to chromatin condensation and transcriptional silencing, or stop of genetic expression. DNA methylation and histone deacetylation lead to stop of gene expression. Condensation of chromatin will be. One more about DNA methylation is connection with aging. Age-related changes in DNA methylation can be different in different places of the genome. In some places, look here in younger people, in the promoter part of genome, there is hypomethylation, global hypomethylation here. But in the elderly people, there is hypermethylation. Methylated bases are with a red color. You can see the red color. And here there is a global and specific, promoter-specific hypermethylation. In the other part of the genome, of the genes, there is hypomethylation in the elderly people and hypermethylation in the promoter part of the genome. The consequence: genomic instability and silencing of genes involved in DNA repair, regeneration, and cell maintenance. The impact on gene expression is that alters which genes are active or silent, contributing to aging-related cellular changes. One more about biological age is testing of one compound which can be hydroxylated after the methylation, which is called 5-hydroxymethylcytosine. After this modification of cytosine to become methylated, by the way, this SAM is the cofactor of methylation, the cofactor of the enzyme. This enzyme, which is called methyltransferase, which uses the cofactor S adenosylmethionine, is because of the transfer of methyl group, and after the methylation of cytosine to 5-methylcytosine, after that there is participation of some enzymes, translocation enzymes, and hydroxylation, additional hydroxylation, and this is the final product, 5-hydroxymethylcytosine, which can be tested. And according to the quantity of this compound, we can understand the biological age, which is different from the chronological age. The key point is that this compound is a dynamic epigenetic mark that complements DNA methylation in controlling gene activity and monitoring the biological age. Other terms except bases, you have to know what are these nucleosides and nucleotides. Do you know the difference between nucleosides and nucleotides? Yes? Yes?
Nucleotides means base and sugar, while nucleosides mean base, sugar, and
opposite. The opposite answer.
Oh.
Yes. Nucleosides means base, pentose. That's all. Nucleotides, base, pentose, phosphate. This is the difference. We start with nucleoside. Nucleoside means base. This is the base. Can you recognize which is this base? You can see the name. If the name is cytidine, what is the base name? Cytosine. It's a cytosine base. And which is this base? If the nucleoside name is adenosine, what is the base name? Adenine. Adenine. Nucleoside means combination between base, pentose. Base and pentose. But how the name is formed? The name in the purine nucleosides is formed by the combination between base name plus ozine. Adenine, adenosine. Guanine, guanosine. The pyrimidine nucleoside name is a combination between base name plus idine. Add idine, and will be cytosine, cytidine, thymine, thymidine, uracil, uridine. These are the nucleoside names. What is the bond between base and pentose? The bond is called beta and glycosidic bond. One more. By which atom from the base there is a connection between the pentose and the base? The answer is, in the purine bases, the bond is between N9 of the base and C1 of the sugar. In the pyrimidine bases, is between N1 of the base and the sugar. This is the difference. There are two conformational forms of nucleosides. Syn, like here, and anti-orientation. The most common orientation is anti. It predominates in naturally occurring nucleosides and nucleotides. Two types of pentoses we have. Two types of pentoses. They can be ribonucleotides in RNA molecules or deoxyribonucleotides in DNA molecules. The difference is only on here, on this second position. In the ribonucleotides there is hydroxyl group. In deoxyribonucleotides no oxygen. That's why the name is deoxy. Deoxyribonucleotides contain deoxyribose, and it is used in DNA. In this table you can see the different names according to the base name. Adenine, adenosine is ribonucleotide. Deoxyadenosine will be deoxyribonucleotide, and etc. Except connected nucleotides in polynucleotide chains, there are some free nucleotides in our cells. And you have to know the role of free nucleotides. This is the first example of free nucleotide, which is called ATP, adenosine triphosphate. In the structure of ATP, we have the base, which is adenine, one pentose, and 3 phosphate atoms. All of these phosphate atoms, each of them has specific name. The first is called alpha phosphate atom, and the corresponding bond is called alpha. The second one is called beta phosphate atom, and the bond is called beta. And the third one is called gamma, and the corresponding bond is gamma. Can you see that beta and gamma are noted with specific symbol like wave? This symbol means that both of the bonds are high energy bonds, both of them. The other word is macrourgic bond. That's why the main role of this free nucleotide is energy donor. Energy donor means that can give energy after the hydrolysis of gamma or beta bond. Alpha bond is not high energy, it's a normal energy bond. Or normoergic bond is the other name. Students, look here. If there is hydrolysis of gamma bond, what will be the product? Can you understand? Which will be the product? This is the ATP. And after the hydrolysis of gamma bond, which will be the products? Can you understand? There will be ADP plus P. These will be the products. Two products. Adenosine diphosphate plus 1P, one phosphate group. This is the hydrolysis of gamma bond. If there is hydrolysis of beta bond, the products will be? Yes, AMP plus pyrophosphate is the name of this compound. Pyro means two, but inside the pyrophosphate there is one bond, which is gamma. Gamma bond is located here inside, and after the additional hydrolysis of gamma bond in this pyrophosphate by the enzyme pyrophosphatase, will be produced two separate phosphate atoms, and more energy will be released in these reactions. All these reactions are exergonic reactions, which means there is releasing of energy in the environment. Remember that ATP is an energy donor and it is used in all reactions which require energy. These reactions that require energy are called endergonic, because exergonic means production of energy. If we're talking about only a base plus pentose, this compound will be only adenosine, which is nucleoside. Other example of free nucleotide is this combination between vitamins and nucleotides. It's a complex compound, which the name NAD or nicotinamide adenine dinucleotide, this one on the slide, or NADP, which is nicotinamide adenine dinucleotide phosphate, if instead of X there is a phosphate group, and some others like FAD and coenzyme A. Let's think about these compounds. Students, NAD, NADP are absolutely different compounds. Don't confuse both of them. They have different participations in the reactions. Let's see what is the action of NAD. NAD is used as a cofactor of dehydrogenases enzymes that take hydrogens from the substrate. These enzymes are called dehydrogenases. For example, if the substrate is this one and the product is this one, what is the type of this reaction? Reaction type. I said the reaction is called dehydrogenation because these two hydrogen atoms need to be taken from the substrate. But the compound that accepts these hydrogen atoms can be NAD plus. This compound accepts the two hydrogen atoms and is converted to NADH plus H plus. These are now these two hydrogen atoms. This is the action of NAD. With other words, NAD is a cofactor of dehydrogenases. Absolutely the same role is about FAG. Instead of NAD, we can write FAG. But the reduced form of FAG will be FAGH, the same. NAD and FAG are oxidized form. NADH and FAGH are reduced forms of both of the compounds. But NADP is used as a cofactor of reductases, another enzymes which catalyze the opposite reaction. For example, this is the substrate, and this is the product of the reaction. The reaction type is a reduction. Reduction. This is the opposite of this one, which is dehydrogenation. The total name, the common name of all of them is oxidoreduction. Dehydrogenation is equal to oxidation. This is the same. This is about the first types of reactions. The second type of reaction is reduction. That's why the enzyme name will be reductase, which is the cofactor of reductases. The cofactor of reductases is NADH, but the written form is NADPH. NADPH. Don't forget P. Don't forget P. It's another cofactor. NADPH enters. NADP is released. NADPH comes from PPP, pentose phosphate pathway, is the name of this pathway which produces NADPH. To summarize, NAD, FAG, cofactors of dehydrogenases. NADPH is a cofactor of reductases. Both of the types are called oxidoreductases, but you need to specify where exactly they use. Not only oxidoreductases. You need to know in which direction they are used. The next example of free nucleotides are all the rest, by the way. Uridine nucleotides are used in the carbohydrate metabolism. For example, UGP glucose. Cytidine nucleotides in the lipid metabolism. Guanosine nucleotides in gluconeogenesis and protein synthesis. And look, this one is very, very important. ATP-ADP ratio. What is this? Students, ATP, you understand that ATP is a free nucleotide, but not only ATP. The ratio is very, very important. Our cells, they react not about the individual quantity of anything. They react of ratios. What about this ratio ATP-ADP? If there is high level of ATP, this means that there is enough energy. This means the cell says, I have enough energy. If there is high ATP and low ADP, enough energy for the cell. If it's opposite, low ATP but high ADP, this means that cell says, I don't have energy. Which processes produce energy? These processes that produce energy are called catabolic processes, or processes of degradation of the compounds. For example, you eat some food. After the eating of this food, everything is degraded to monomers, and during these catabolic degradation processes, energy is produced. Now, ATP is increased and you have enough energy. You stop eating. But because you hear, I have enough energy, you hear yourselves. But if you continue eating, ATP will be produced more and more and will go to synthetic processes, and you can gain your weight if you cannot hear yourselves. That's why ATP-ADP ratio is the most important for the regulation of all catabolic degradation means and anabolic synthesis means, processes. Of course, it's not the only ratio. There is ratio between insulin, glucagon, well-fed or starvation state, and etc. But now we're talking about the free nucleotides. That's why remember, if you cannot answer of any question about the regulation of any enzyme, your first answer need to be HP-ADP ratio. This is the first answer for all enzymes. These processes that produce energy are catabolic ones, and these are two of them: oxidative process, mitochondria, and glycolysis. These nucleotides that act as positive allosteric regulators means that they activate. These that act as negative regulators, this means that they inhibit. That's why the second option is allosteric regulation. And one more example of free nucleotide is this one. It is called cyclic AMP. It can be produced from ATP by the enzyme adenylyl cyclase. One very similar of cyclic AMP compound is this one, which is called cyclic GMP. What is the action of both of them? These cyclic nucleotide students, the main types, are used as second messengers in the signal transduction. What is this signal transduction? It's a process of transmission of information from outside the cell to inside the cell. How this information can be transmitted? By hormone action. Because if the hormones are water soluble, they cannot cross through the cell membranes. Do you remember this from the first lecture? Because the cell membrane is lipid soluble, which means only lipid soluble compounds can cross the membrane. The water soluble cannot. That's why the water soluble hormones are recognized from membrane receptors. They cannot enter the cell. They stayed outside the cell, the water-soluble hormones like insulin, glucagon, adrenaline, somatotropic hormone, oxytocin, vasopressin, and etc. Many examples of water-soluble hormones we know. But inside the cell, they transmit the information thanks to secondary messengers. For example, adrenaline and glucagon work with this second messenger, cyclic AMP. Nitric oxide works with cyclic GMP as second messenger, and etc. After this transmission of information, the giving the information from the hormone to the receptor, after that to the second messenger, then there is transfer of information to the corresponding protein kinases. Example, if there is action of cyclic AMP, the protein kinase which is activated is protein kinase A. If there is action of cyclic GMP, the protein kinase which will be activated after that, it is called protein kinase G. ATP, cyclic AMP, protein kinase A. Or GTP, cyclic GMP, protein kinase G. This is the connection. Deoxyribonucleic acid. Something about DNA. First, the human DNA and general characteristics. Number one, the DNA is located in all tissues, in all our cells. And it is the same in all tissues of the individual. It is organized in 23 chromosomes or 46 in somatic cells. What is the length of DNA? 2 meters. 2 meters length. And the nuclear DNA, which is around 99% of DNA, the mitochondrial DNA is about 1% only, which is circular. This one is linear in DNA. And what is the full condensation in DNA? The full condensation is called formation of chromosomes. They are formed only during the mitosis, during the interphases, no chromosomes. This compound is called chromatin. Chromatin. Do you know what is this chromatin? It is a combination between what and what? Yes?
DNA and protein.
Yes, well done. It is a combination between DNA and proteins. And there are two types of chromatin: AU chromatin, which is with possibility with transcription. It's an active one, and heterochromatin, which is condensed chromatin. Watson and Crick model. If we're talking about the Watson and Crick model, I think you know a lot that the DNA is the carrier of genetic information, transforms non-encapsulated pneumococci into encapsulated forms. The Chargaff's rules means that the quantity, the number of adenine bases is equal to the number of thymine bases, or the number of cytosine bases is equal to the number of guanine bases. How many hydrogen bonds there are between A and T? Yes? Two. Between G and C? Three. Yes. Okay. And the connection between nucleotides. What is the name of these bonds between nucleotides, which form the primary structure? Yes? Yes?
Hydrogen.
No. Between nucleotides, not between bases. Yes? Yes, they are covalent bonds, but they are called 3', 5' phosphodiester bonds. These are the bonds. And thanks to phosphate groups, which are located outside, which have negative charges, that's why the total charge of DNA is negative and can be combined, can be attached with some positive charged proteins, for example. But inside the helix are located the bases, which form these hydrogen bonds between them. And this is the primary structure of one DNA. For example, it's one nucleotide which contains cytosine as a base. The next one contains adenine, and the next one contains guanine, and etc. This is the primary structure thanks to the formation of 3', 5' phosphodiester bonds. There is polarity. Yes, and the polynucleotide chains are open, not circular like in the mitochondria. The secondary structure of DNA refers to hydrogen bond formation between complementary bases. The base, so-called base stacking or PP interactions, which stabilize the interaction between adjacent bases, hydrophobic interactions, and ionic interactions with the participation of magnesium, which neutralize phosphate backbone. Actually, magnesium is used for the stabilization of external the phosphate atoms in the ATP, and in all reactions where there is participation of ATP, there is magnesium. Obligatory, there is magnesium. You'll see this. This slide shows that sometimes can be formed thymine dimers. Thymine dimers by ultraviolet radiation can induce covalent bond formation between adjacent thymine bases, and this is called thymine dimers. This formation of thymine dimers, students, is a mutation in DNA, and the clinical relevance leads to impossibility of DNA repair mechanisms, which result in this molecular disease. Molecular disease, that means that there is impairment of some nucleotides in some codons in DNA, which can be influence the formation of enzymes, transport proteins, or any proteins that are involved in the organism. And this is characterized with sensitivity of the skin to sunlight. It is called also photosensitivity. And the disease is called xeroderma pigmentosum. This means that these people cannot go out during the day. They need to stay at home, and just during the night they can go out of their home because of the sunlight. Because after, if they under the sunlight, it will be increased risk of malignancies in their body. There are three types of conformation of DNA. The most common DNA is B form, which is the most stable and predominant form of conformation under physiological conditions. It's a right-handed helix with 10 base pairs per turn, this one. And the base pairs oriented perpendicular to the helix axis. Actually, this form is described by Watson and Crick. aDNA, this one, is again right-handed, but with 11 base pairs per turn. And this one, Z form, is left-handed with 12 base pairs per turn. It's also so-called zigzag-shaped backbone. The common between all these double helices is that the chains are anti-parallel and complementary. This is the common. And actually, the biological role, what is the biological role? Transitions between conformations may contribute to regulation of gene expression. Packaging. The question is, how is possible this long DNA to be packaged, to be included in this small nucleus? How is possible? Of course, with strong packaging, with strong condensation, and the first level of condensation is participation of histone proteins. Histone proteins are basic, positively charged, that bind tightly to DNA because of its negative charge that comes from the phosphate groups, which are located outside the helix. That's why the histone proteins can be contact with the DNA. And the first stage is formation of nucleosomes. Students, except histone proteins, there are many non-histone proteins which are diverse group, interact with DNA, and the function is structural support of chromatin and regulation of gene expression. For example, some of these non-histone proteins are transcriptional factors. And let's see the formation of histones, of nucleosomes, thanks to the histones. This is the example of one histone. These are actually H2A, H2B, H3, and H4. Each of one is multiplied 2. Total number 8 histone proteins they are involved in one molecule of nucleosome. Between two adjacent nucleosomes, there is formation of histone H1, which is located in so-called linker part of DNA. And the 147 bases are wrapped around the DNA in this histone octamer. Octa means 8, yes. And after this formation, the DNA will be around, the DNA fiber will be around 10 nanometers from native structure, which is 2 nanometer, thanks to nucleosomes, will be 10 nanometer. And after that continues the condensation, and the full condensation, I said, will be during the mitosis with the formation of chromosomes, only during the mitosis. In the beginning of the lecture, I said something about DNA methylation and histone deacetylation. Do you remember that I said this leads to chromatin condensation? Let's see something about histone modification. Histone proteins, we said that histone and non-histone proteins are involved in the structure of chromatin. They are together, but histone proteins can be modified. This modification is about some amino acids that are involved in the structure of these histone proteins, and these amino acids can be modified by acetylation and deacetylation. Acetylation means that the compound that is used for acetylation is called acetyl coenzyme A. This process of acetylation means contact with acetyl group. Deacetylation means removing of acetyl group. And if there is acetylation of histone proteins, there is transcription activation with relaxed chromatin, which means active. If there is deacetylation by enzymes called deacetylases, the chromatin will be condensed, and this means transcriptional repression. And students, there are specific drugs that are called histone deacetylation inhibitors. They are used for anti-cancer agents because can restore expression of tumor suppressor and pro-apoptotic genes. Because if there is inhibition of this process, they will become active, which means if we're talking about the tumors, they will activate the pro-apoptotic genes. Apoptosis means program cell death, which means will lead to the degradation of tumor cells in the end. These drugs are used as anti-cancer agents. Acetylate histones and non-histone proteins can be for different proteins that are involved in these signal transduction pathways. And the effects in the end will be cell cycle arrest or induction of apoptosis or anti-angiogenic activity. What is this angiogenesis? Angiogenesis means formation of new blood vessels in the cancer cells, in the tumor, because they create their own blood vessels. If there is anti-angiogenic activity, means to stop the formation of new blood vessels, and this will reduce the tumor formation. DNA denaturation and renaturation. Students, don't confuse DNA denaturation with protein denaturation. Again, it's denaturation, but for other compounds. DNA denaturation means that hydrogen bonds between complementary bases are disrupted, but not 3', 5' phosphodiester bonds, which are covalent bonds are very strong bonds. It's similar to the peptide bonds in the polypeptide chains. Yes, and two terms you need to know about denaturation. The first one is so-called hyperchromic effect. What is this hyperchromic effect? And what is this? Melting temperature. Melting temperature is this temperature when there is 50% of denaturation of a DNA molecule. Exactly this temperature is called melting temperature. When the melting temperature will be high and when will be low depends on the quantity, the quantity of the AT pairs or GC pairs. What does it mean? If there is a low melting temperature, this means that there is with low stability, it's a low stability DNA. And what kinds of pairs will be rich? It will be rich of AT pairs with low melting temperature, or rich of GC pairs. What do you think? Low melting temperature means that the DNA is rich of adenine-thymine pairs. Why? Because they are connected between each other with only 2 hydrogen bonds. If there is GC rich pairs, this means that the DNA is more stable. Why? Because the hydrogen bonds between guanine and cytosine are 3, that's why. And what is this hyperchromic effect? Single-stranded DNA absorbs more UV light at 260 nanometers than double-stranded DNA. Which means if there is increasing the absorption of UV light exactly at 260 nanometers, this means that exactly at this moment there is increasing of denaturation of DNA. Because if we have single-stranded DNA, this means denaturated DNA. And under appropriate conditions, complementary strands reform again the double helix. Students, exist some synthetic nucleotides analogs which are used in chemotherapy. And there are 2 types because there are 2 mechanisms of action of these synthetic analogs. The first mechanism of action is inhibition of some enzymes which are essential for nucleic acid synthesis. This is the first mechanism of action. Let's see what I mean. It's students about this example. The example of this drug, which is a synthetic analog, is called allopurinol. It's a purine analog. Let's see the structure and you understand. This is the allopurinol. This is the structure of allopurinol. What is this actually? It's absolutely very similar to well-known purine bases. That's why we can say allopurinol is a structural analog of what? It is a structural analog of one base which is a part, it's an intermediate of purine, not biosynthesis, sorry, in the purine degradation. In the purine degradation. I said about that today. This base is not from the common bases. It is called hypoxanthine. Hypoxanthine is one of the intermediate of purine degradation. Do you remember that I said something about gout? Hypoxanthine goes to xanthine, and in the end uric acid is produced. If there is inhibition of this enzyme, which is called xanthine oxidase, no uric acid in the end will be. Allopurinol is used exactly in this reaction. It inhibits this reaction, or it inhibits the xanthine oxidase enzyme, which catalyzes both of the reactions, in the first and in the second reaction, both of the reactions. And after that, students, no uric acid in the end. But why can inhibit the xanthine oxidase? Because it's a structural analog of hypoxanthine, that's why. And this is the first mechanism of action. Inhibition of enzymes which are essential for nucleic acid synthesis. The second action is incorporation into DNA or RNA. I'm talking about the synthetic nucleotides. They can be incorporated into DNA and RNA, but which DNA and RNA? In the tumor cells. That's why these types of synthetic nucleotides, like 5-fluorouracil, or 3-deoxyuridine, or 6-mercaptopurine, or etc., because of their incorporation into DNA prior to cell division. This leads to inhibition of tumor growth, which means they are used in the cancer therapy. And you can see different, absolutely different examples as structures. And in the end, something about RNA, ribonucleic acid, but only from biochemical point of view, with no other details. First, I think you know the differences between DNA and RNA. You know that the DNA is a double-stranded molecule with a long chain of nucleotides. RNA is a single-stranded molecule with shorter chain. You know the participation of adenine and thymine, but here is adenine and uracil in RNA. The DNA in nucleus and mitochondria, RNA in nucleus and cytoplasm, and the difference in the pentose, deoxyribose, ribose, transmission of hereditary information, and here all RNA are involved in the protein synthesis. And this is the primary structure of RNA. And in this slide you can see different examples of RNA, but not all, because exist many types that are not written here in this slide. Actually, here you can see only the main types of mRNA, which carries genetic information from DNA to ribosomes for protein synthesis, tRNA or transfer, which transfers specific amino acid, and RNA, ribosomal RNA, which form the core of ribosomes, but together with proteins, don't forget again, and some other small types, like small nuclear RNA, microRNA, small interfering RNA, and there are many other types of RNA. Something about all of them, the main types only. Messenger RNA, 5%, and the most important of these, if we're talking about the synthesis, is that in the beginning of synthesis of mRNA, there is formation of one precursor of mRNA, is inactive, very big form of mRNA, which undergoes the process of maturation. This process of maturation requires formation of spliceosome, and let's see something one more, what there is in the 5' end and in the 3' end. This is important to know. In the 5' end there is so-called cap. This cap is formed by the formation of 7-methylguanosine triphosphate. The link is 5' triphosphate linkage. And in the 3' end there is formation of so-called poly-A-tail. Poly-A means formation of many adenine bases. How many are these bases? They are from 40 to 250 adenine bases in the end. What is the role of this additional post-transcriptional modification? It's because of the for the protection. Cap and tail are for the protection of mRNA, and during this post-transcriptional modification of mRNA, there is this splicing, which means removing of introns and the joining of exons in the end, and the mRNA is mature, is ready to use. The transfer RNA, students, at least one specific per amino acid. The most important is that in tRNA there are many modified bases, by the way, which means methylated bases, a lot, and the function is they carry specific amino acids, recognizes the codon on mRNA, and this is the interesting secondary structure at cloverleaf model with different loops And in the end, ribosomal RNA, which there are 4 species in eukaryotes, 28S, 18S, and 5.8 and 5S, or Svedberg units. This is the 80% of total cellular RNA, and don't forget that the formation of the ribosomes requires the combination with specific proteins. If you remember, during the first lecture, I said something. We talked something about the proteins that are used for the stability of the environment, and we set example of collagen, which is used with structural function in the connective tissue. But don't forget these proteins that are involved in the formation of ribosomes, because they have the same structural function in the formation of ribosomes. And don't forget that some RNA molecules have catalytic activity, and they are used ribozymes. Ribozymes means enzymes. We know that most of the enzymes are proteins, but some part of them are called ribozymes. And is the last slide, which shows the formation of ribosomes. The combination, again, between rRNA, and look how many proteins are involved here or here. Many proteins which have the function of structural function. It was everything for today. Thank you for your presence.